3 lectures to go. So please, as a reminder, subject evals are posted. If you have time to do those, please do those. We can start here with the polling question. Last time we talked about a lot about fatty acid synthesis. Question here in the polling question, fatty acid synthesis. Okay, good. Most of you have it right. Perfect. And then the other thing I talked about last time, I just quickly summarized up here. We also talked about fatty acids. Of course, they generate acetyl-CoA. I told you last time, every time you synthesize a fatty acid, adding a 2-carbon unit after the first acetyl-CoA. So the first acetyl-CoA doesn't cost anything other than making the first acetyl-CoA. But the first acetyl-CoA costs you 1 ATP and 2 NADPH to add those. And of course, you can also break down fatty acids, we learned, to acetyl-CoA, and that can be involved in cholesterol and isoprenoid synthesis. It's called so-called mevalonate pathway, which I went through last time. I alluded to HMG-CoA reductase being the target of statins, one of the most common drugs that you future doctors will prescribe to people or that you may take in your lifetime. Also, we talked about that, remember, one of the consequences of the fact that we can't turn anything 2 carbons or less into glucose means that if you're living off of just fat, the so-called true ketogenic diet, that you can then get in a situation where if you can't maintain blood glucose, you'll switch over to this ketone metabolism, where acetyl-CoAs can be combined to make acetoacetate, which is in equilibrium with beta-hydroxybutyrate, and these 2 molecules being the so-called ketone bodies. That's an alternative fuel source for the body versus glucose and pyruvate, lactate, that sort of fuel source system. Okay, so today, and really is the first of the final 3 lectures that are about nitrogen metabolism. So this time and next time, I think I titled them amino acid metabolism 1 and 2, and the last one's nucleic acid metabolism, but it's really all intercombined. And you could probably guess we drew nitrogen an awful lot of times during this class, both my half and Professor Yaffe's class. And nitrogen is, of course, essential for life. You need nitrogen to make amino acids, you need nitrogen to make nucleic acids, and it turns out that nitrogen metabolism is really about the synthesis and breakdown of nucleic acids and amino acids and how they relate to the physiology that handles nitrogen by living organisms. Now, up till now, the entire focus, or much of the focus of sort of my half of 705, the metabolism half of 705, has been about sugar and fat, how we make sugars, how we break down sugars, make fats, break down fats, because largely that's about energy transduction in cells. But if you think about it, we eat other organisms. We have to. Life requires photosynthesis, so we have to eat other organisms in order to get the energy to live. And what are organisms made out of? They're mostly made out of protein. There's some nucleic acid there. So all of us, whether we're intentionally eating a lot of protein in our diet or not, we eat a lot of protein. The first half of the course, Professor Yaffe, what he largely taught, was largely about how proteins and nucleic acids allow cells to work. And proteins and nucleic acids, as I alluded to, all contain nitrogen, and nitrogen becomes very important for those molecules, but of course they have to come from somewhere, and that's really what we're going to focus on for the last 3 lectures. Now, most of you, if you think about I'm going to eat a lot of protein, it's usually about how am I going to gain muscle mass, right? Anabolic. There's weightlifters out there. I made comments about this earlier, talking about like creatine and stuff like that. If you go to a nutrition store and you look on the side, the bodybuilding supplements, they have things like leucine in it. We can debate what those things do, but I will tell you that there's a reason why people buy that stuff, and it's because if you want to build muscle mass, yes, you have to work out, but you also need nitrogen. And so things like creatine, leucine, things like that have nitrogen in them that are necessary to do this muscle building. Now, we'll also point out that a major function of the muscle in your body is actually to store amino acids. You don't think of it this way, but your body is constantly breaking down and building muscle. in every single one of us every single night, because this is a place that we store amino acids. In fact, if you stop eating, okay, completely stop eating, what happens? Well, you're going to burn up all the sugar that's in your blood to begin with, and then the liver is going to switch to glycogen, and muscles will break down glycogen, and then ultimately you're going to run out of glycogen. So then what happens next? Well, you can turn to fat, but before you fully switch over to burning fat and ketone metabolism, you'll also start breaking down muscle. You'll break down muscle because you can use amino acids for gluconeogenesis, which is why those of you who think you're doing the keto diet just by eating low sugar, you're not. A true ketogenic diet, you also have to have low protein, because the amino acids in protein can be used for gluconeogenesis, as we'll talk about, I think, next time. So a true ketogenic diet is actually quite gross. It's just fat. It's eating like a stick of butter, because that's the only way you can avoid something else that can be made into glucose. But the bottom line is all of us can live off of our muscles for a very long time as well. We actually have quite a lot of energy stored in our muscles because we can use that amino acids there as fuels and as things to do like gluconeogenesis. But if we're going to burn amino acids, and by the way, there's like, I don't know, I didn't get around to making an enrichment lecture about this, but happy to talk about the office hours or something like that. So you guys see in the news like GLP-1 agonists, right? So GLP-1 agonists, great weight loss drug. Actually, for the first time, weight loss drug that actually works and is relatively safe. But there's a downside to every single medication, and the downside of the GLP-1 agonists is people lose weight, but they also lose muscle mass, which is not always best for their metabolic health. And part of that is because what they do is they suppress appetite. So you eat less, you start breaking down your muscle as well. And there's really not a whole lot you can do about that, because that's normal physiology. Although certainly lots of people are looking for other weight loss solutions that preserve muscle mass. But regardless, if you're going to break down muscle or eat other organisms and use amino acids for food, you have to deal with the nitrogen, right? The nitrogen has to go somewhere. And so this is really where we get into amino acid metabolism. And we alluded to this when we talked about the malate aspartate shuttle, but it turns out all of our amino acids are related to alpha keto acids. So if you think back to the malate aspartate shuttle, and I'll draw these for you in a little bit, remember it involved oxaloacetate, which ended up being interconverted with aspartate, right? I told that Professor Yaffe harped into the 1-letter and 3-letter abbreviations for amino acids. All right, so I'll remind you of some of them as we go along here, but I will use them. So aspartate, D-aspartate, okay? The other one, as part of the malate aspartate shuttle, was alpha ketoglutarate and glutamate, okay? Glutamate, single letter E. So I'm not going to draw those out right now, I'll draw them out later, but if you draw them out, the alpha keto acid, alpha ketoglutarate, if you switch the ketone group for an amino group, you get glutamate. Oxaloacetate, another alpha keto acid, also beta keto acid, that's not drawn up there, but alpha oxaloacetate, swap the keto group for an amino group, you get aspartate. I'll draw another one just to drill the point in. Here's the amino acid. Okay, so this here is the amino acid alanine. Okay, alanine, A-L-A-A. All right, that, if I swap out this amino group for a keto group, get a molecule I've probably drawn 500 times over the past 5 weeks, pyruvate. Okay. Alpha keto acid pyruvate, amino acid alanine. All right, so if we can basically chemically convert keto groups, alpha keto groups, for amino groups, that tells me how I can interconvert nitrogen containing molecules, amino acids, for something that I can burn to get energy. Obviously, we talked at length. Pyruvate, you want to burn in the TCA cycle, do the pyruvate dehydrogenase reaction, get acetyl-CoA, put it in the TCA cycle, turn it into CO2. Same thing, I can make oxaloacetate or alpha ketoglutarate, put it in the TCA cycle, I can burn it to CO2, make lots of NADH, pump protons, do oxidative phosphorylation, get ATP, get heat, whatever else we want to do for energy. But if I'm doing this, if I want to burn these things to CO2, I gotta put the nitrogen somewhere, right? The nitrogen has to go somewhere. It also follows then that if I want to do the opposite, if I want to build amino acids, I want to build muscle, I want to make protein, amino acid has to come from somewhere. Well, you know the reactions I can use to make pyruvate, and oxaloacetate, and alpha-ketoglutarate. And if I have a source of nitrogen from somewhere, if I can do this reaction, I can put the nitrogen on, and now I have a way to make amino acids that I can use to build protein. And so how cells basically deal with nitrogen, bring nitrogen into the system, making amino acids, nucleic acids, take nitrogen out of the system to burn amino acids, is really nitrogen metabolism. And so talking about nitrogen metabolism, breakdown and building of amino acids, and it turns out also nucleic acids, is largely about how organisms handle nitrogen. So to get at this, I have to say, well, what is biological nitrogen and where does it come from? So if you want nitrogen right now, it's pretty easy to find. It's all around us, right? Nitrogen, most common gas in the atmosphere, all right? That's great. We also eat a lot of organisms. If we eat a lot of organisms, we can get nitrogen from eating other organisms, because even if I'm totally vegan and I eat plants, only plants, my plants that I'm eating, those are still 50% protein, the cells in there, roughly 50% protein. So I'm getting a lot of nitrogen just from that protein. So it turns out heterotrophic organisms, all of us, things that eat other organisms that don't do photosynthesis, all of us don't actually have a nitrogen problem, or we do, but our problem is getting rid of nitrogen, because everything we eat is 50% nitrogen containing, and that's excess, that's more than we need, so we need to get rid of it. But if you're a photosynthetic organism, you have the complete opposite problem, okay? If you're a photosynthetic organism, now you can build everything from scratch. You can take gas from the sky, CO2, turn it into a plant. But you need nitrogen because you're 50% protein, so you're always needing nitrogen to build that protein. What's fertilizer? Like a garden or something like that? Fertilizer makes a difference. What's fertilizer? Mostly nitrogen, also phosphorus, but phosphorus and nitrogen, those are the main things that are in fertilizer. So ultimately life, because life evolved from something that can do photosynthesis, had to get nitrogen from somewhere, and that nitrogen is in the gas that's all around us, okay? So let's think about it. What's nitrogen gas that's around us? It's N2, all right? So if I draw this out, remember nitrogen has 5 valence electrons, okay? So that's Lewis structure of N2, nitrogen gas. This is pretty chemically inert for any of you out there who are course 5 or course 10 or whatever, right? So you go to your lab, right? What do you do to stabilize stuff? You spray nitrogen over it because it's cheap and it's inert and it'll stop things from reacting, unlike that terrible oxygen, which we talked about really likes to rip electrons off of things, okay? So nitrogen's pretty inert, so this is pretty useless to things. What do we have to do in biology in order to make this not inert? So what is in amino acids and proteins and nucleic acids? It's basically amino groups. So what's an amino group? So if I draw the Lewis structure here of this, okay, this is basically, I've taken that nitrogen and I've now added 3 electrons, okay, and I've added 3 protons. Now I have NH3, ammonia, okay, that's not inert. That's effectively what we're dealing with in proteins, okay? Clean the floors with this, it kills things, smells bad, okay? Ammonia, not inert, nitrogen gas is inert. So effectively, for biological nitrogen to be useful, I need to add 3 electrons, or I need to reduce my nitrogen to a more reduced form of nitrogen, ammonia, and this is now very useful for life in biochemistry because now I have this amino group that I can put into proteins, amino acids, nucleic acids And if you think about this, you've been looking at nitrogen participating in reactions involving life throughout this entire class, right? What makes all those hydrogen bonds that give your protein secondary structure? What makes all the hydrogen bonds that allow you to do the base pairing in nucleic acids? Active sites of enzymes that I've shown you, okay? So getting to this nitrogen reduction, okay, so-called nitrogen fixation, which is basically reducing N2 plus balances for you, 6 electrons plus 6 protons going to 2 ammonia molecules. Okay, that reaction, nitrogen fixation, nitrogen reduction, is probably one of the most important reactions that happens on Earth that allows life to exist. Life would not be possible without two things: photosynthesis, which we already talked about, and nitrogen fixation. And this is probably the most important topic that we don't really cover all that much in 705, mainly because we don't have time. But effectively, nitrogen fixation is really only carried out by a very limited number of organisms on Earth, the so-called cyanobacteria. If you wanted to eliminate life on Earth, if you killed all the cyanobacteria, we'd be screwed. Okay? It's basically one set of bacteria that allow nitrogen fixation to happen. And if you care about things like environmental science and whatnot, these are actually really an important thing. You can read about this. It's complicated redox chemistry. It's super expensive to carry out these nitrogen fixation reactions. It's like 16 ATP, as well as all this reduction that's happening, reduction, energy input, all that other stuff. So very few organisms can do it. Why do very few organisms can do it? Because we have a lot of biological material out there, and so most of us, the issue is getting rid of nitrogen. But if you care about, say, carbon fixation from the atmosphere to store CO2 in plants, dealing with carbon fixation is a big deal. I'll also say, like, if you read in the news, like, why is, like, fertilizer use problematic when it, like, runs off into water sources and stuff? Effectively, because you're flooding water sources with nitrogen, that causes algal blooms, which can be toxic to other organisms. And so lots of environmental science is kind of tied up within things like nitrogen and how that interacts with various photosynthetic organisms. So anyway, I just want you at a minimum to be aware of this as a problem. But regardless, biological nitrogen, which is effectively ammonia or its conjugate base, ammonium, all right? These are two molecules, because as I said, like, you guys probably know what this smells like because it's cleaning solution, right? It's toxic, it's nasty, all right? This is not something that typically you like to have in high concentrations, yet this is essential for life. So in small quantities, this is good. In large quantities, this is bad. Yet us as animals, we eat other organisms. We need to get rid of nitrogen, which ultimately means we need to get rid of ammonia. Now, different organisms out there will eliminate nitrogen in different ways depending on their lifestyle. And the simplest strategy is simply excrete ammonia. All right, so if you're a fish or some other aquatic organism, I can excrete ammonia just out into the ocean, right? The ocean is big, it's dilute, right? What's the old saying, right? You know, you're pissing in the ocean, right? What does that mean? It just, doesn't matter. It's, you can pee in the ocean, it won't bother anybody. Well, fish do that all the time. They're releasing ammonia directly out into the ocean all the time because it doesn't matter. The ocean is big and dilute, it's not an issue, all right? However, terrestrial animals, us, need a different solution. All right, we don't live in water, okay? We can't just put stuff out into the ocean. So what's our solution? So our solution is we generate urine, all right? This is not really. I had to do this years ago, so Mike Yappy didn't do this year. It's too bad for you guys. He usually draws his blood. Have you guys heard that before? So when Mike draws his blood and that like creates this big stir, I have to think about what biological fluid could I bring in to be relevant for 705. I don't know, it's like stale beer juice or something. But anyway, if you want a demonstration, you guys, every time you urinate, you are excreting nitrogen, okay? So what are you excreting in there? It's basically water control, but it's also nitrogen. And what the main reason you make urine is to get rid of this molecule, which is urea, all right? So your strategy and other terrestrial mammal strategy basically take ammonia, turn it into urea, which also requires a fair amount of water to get rid of, to excrete in urine. And this is why we as humans need to urinate several times a day, because that's effectively our nitrogen excretion. However, to get rid of urea requires a fair amount of water, okay? This is pretty concentrated urine that I showed you right here. Your urine color will go up and down based on how much water you have, but there's a limit, right, to how much is there. If you've ever worked in a protein biochemistry lab, you use urea all the time because high concentrations of urea will denature proteins, right? It's not totally benign molecule, so you need a fair amount of water to get rid of it. Some animals that don't have access to a bunch of water will use yet a different solution. So reptiles, largely living in the desert, they actually don't make urea. Same thing with birds, right? Birds don't want to fly around. They need to be light enough to fly. They don't want to carry around a bunch of extra water in order to excrete urea. So instead, they actually generate this molecule. Okay, so this molecule is uric acid. Looks a lot like the purines that Professor Yappy drew for you, because it is a purine. It's actually the most oxidized purine you could possibly get. And uric acid is how reptiles and birds excrete their excess nitrogen. So all of us have experienced bird poop, either because we found it on our car or it fell on our food at a picnic sometime or something like that, right? Bird poop doesn't look anything like human waste, because birds don't make the same waste we do. Bird poop is a slurry of basically solid waste from the bird, whatever it eats, same as your stool, plus uric acid. So birds don't urinate, birds make bird poop. They eliminate their nitrogen waste as uric acid in the slurry that is bird poop. And you can see there's 4 nitrogens in it, so if you can synthesize purines, turn it into uric acid, that's another way to eliminate nitrogen. And so you have all these options for nitrogen elimination. So in the ocean, ammonia; for us, urea; for birds, uric acid. Depending on your lifestyle, your water choices, what's out there, what environment you live in, you can use these different choices. And I think this really illustrates that how we as life deal with our nitrogen, all of us who eat things have to deal with this, choose basically different ways to do it, because effectively how we handle nitrogen has to do with how we generate all kinds of different things that can make these various waste products. In other words, the interconnection between ammonia, amino acid metabolism, urea, which you'll see is tied to amino acid metabolism, uric acid tied to nucleic acid metabolism, is really about how nitrogen is handled. Okay. So before we get into some specifics, one other point I want to make about humans, which is kind of interesting, but also helps illustrate an important point about amino acids. And that is, so we're heterotrophs, okay? Every single one of us has to eat other organisms or we can't live, which means we have to get rid of excess nitrogen in our diet, okay? You eat a ton of protein, okay? If you're good out there and eat the high protein bars and all the other stuff, the thing that's the current view of what's healthy, all right? You have more nitrogen to excrete. Okay, if you're really bad and eat nothing but pasta and bread and not a lot of nitrogen, you're still eating nitrogen, you excrete less nitrogen. So how much nitrogen you excrete basically has to do with how much nitrogen you take in. All right, so if you're healthy, it turns out that the nitrogen you take in is greater than the nitrogen that you put out in the urine. Okay, so there's tests that you can order in the hospital that's like a 24-hour nitrogen in the urine. And if you measure all the nitrogen that you eat, the nitrogen you take in is going to be greater than the nitrogen you put out. And that's partially because you think, oh, you're growing and stuff like that. But even if you're not growing, this is still true, and that's because, of course, you don't account for anything. Some nitrogen is lost in the stool and stuff like that. But this thing, so-called positive nitrogen balance, is actually a sign of health, okay? Turns out that for you future doctors out there, there will be conditions that your patients are in where the nitrogen in is less than the nitrogen out, so-called negative nitrogen balance. That is bad, right? That is effectively seen in all kinds of really chronic, terrible disease conditions, so-called cachexia, right? Losing muscle mass and things like that. Happens with cancer, but also happens with like sepsis, overwhelming infection, HIV, things like that. HIV that progresses to AIDS, not well-controlled HIV. All of those things, negative nitrogen balance is the thing. Now, this is not measured so much in the hospital anymore, although it happens sometimes. Although in the 1950s, people like to experiment on med students. So I know some of you want to go to medical school. So be glad you're born in, or whenever you were born, it's 2025 or 2027 when you go to med school. Because in the 1950s, your professors would have experimented on you, right? And it turns out what they did in the 1950s, it was at Harvard that actually did this, they actually made diets and made medical students, I doubt they had a choice, actually eat diets that were deficient in every, one at a time of every single one of the 20 amino acids that were found in protein. And it turns out when they did this, a bunch of those diets actually led their med students to be in negative nitrogen balance, right? Students got really sick just by dropping out one amino acid from their diet, okay? Some amino acids mattered, some didn't. Now it turns out if you're a prokaryote, you're a plant or a yeast or a bacteria, if you have enough energy and you have a source of nitrogen, of ammonia, symbiotic relationship with a cyanobacteria, or you are a cyanobacteria, or you eat something that gives you ammonia, it's fine. You can actually turn that ammonia into all 20 amino acids you need to make proteins. But you as humans cannot do that, okay? So what they learned from these experiments is that turns out that there are 9 amino acids for you that are essential, meaning you have to eat them from the diet, and there's another 2 that are conditionally essential. And that is, you can make them, but you need to start with an essential amino acid to make them. So what does that mean? That means of the 20 amino acids that are out there that you need to make protein, 11 of them you have to get from the diet. And so 11 of them, when they were eliminated from the diet of medical students, made the medical students sick because they didn't have a way to actually make those. And shockingly, that's how this was actually discovered. And so I listed them up here. I'll write them here before. I'm going to use they're written out. But the essential amino acids are lysine, K, histidine, H, branch chain amino acids, isoleucine, leucine, and valine, threonine, tryptophan, W, phenylalanine, F, and methionine, and the 2 conditionally essential ones are tyrosine and cysteine. So you can make, remember, phenylalanine and tyrosine, hydroxyl group on the tyrosine, pretty closely related. You can make tyrosine, but only if you have a source of phenylalanine. These are the 2 sulfur-containing. You can make cysteine, but only if you have methionine. All right, so all of these amino acids you have to get from your diet. Now, this is not really an issue unless there's some demented professor in the 1950s who's taking these away from your food. All right? It's not really an issue because, like I said, everything you eat is 50% protein. And so that 50% protein, you have plenty of these amino acids, all right? The bigger issue for you is nitrogen excretion. And it turns out the other 9 that aren't on this list are ones that you retain the ability to make. You retain the ability to make them largely because they're involved in other aspects of metabolism. They interact with metabolism in other ways, which will be clear. Okay, so the way you deal with ammonia, okay, so ammonia is really your base biological nitrogen. This really comes in and out of biology via 2 amino acids, glutamate, wrote it up there, and glutamine, Q-tamine, all right, glutamine and glutamate. Those are sort of the 2 amino acids that you really use to put ammonia in and out of biological systems, all right? So, for you. Okay, so here, as promised, here's our old friend, alpha-ketoglutarate. Okay, 5-carbon alpha-ketoglutarate from the TCA cycle is an alpha-keto acid. All right. Now I come along here with my ammonia. This intermediate, and now if I come along and I reduce this carbon, turns out I could do this with NADH or NADPH, okay, both of them, as you've now seen a million times, will give you a hydride ion, okay, and what will that give me? That'll give me okay. I've changed the keto acid, alpha-ketoglutarate, to the amino acid glutamate. E-glutamate. Okay? So the enzyme that carries this out is an enzyme called glutamate dehydrogenase. It's a dehydrogenase because the amino acid, the carbon is more reduced than the alpha keto acid. Okay, in one case it's the ketone, the amino group is more reduced. So there's an oxidation reduction reaction. Turns out glutamate dehydrogenase, which is sometimes abbreviated GLUD, glutamate dehydrogenase, can use either NADPH or NADH, or NADP or NADP, depending on direction. This is completely reversible, and it's basically, what does that mean, reversible? Delta G naught prime is approximately 0, so it can take nitrogen in or out of the system, depending on putting need to put nitrogen in or you need to pull nitrogen out. And it turns out via this reaction, glutamate dehydrogenase, I guess more than any other, is the major reaction that takes nitrogen from ammonia in and out of biological systems. Okay, so that's glutamate dehydrogenase. The other major nitrogen carrier, glutamine, is an important nitrogen carrier for your body because it's also a way that you deal with ammonia in your tissues. Okay, so you have 2 reactions that, or 2 enzymes here that are involved with basically making and breaking down glutamine. Okay, so the first one is glutamine synthase. Okay, so what is glutamine synthase? All right, we're going to start with glutamate. I'm not going to draw it again, it's drawn up there. And if I come along and I phosphorylate glutamate, and I'm going to phosphorylate it on that, basically the R group carboxylic acid. Okay, so phosphorylated glutamate, now my amino group can just come in and displace the phosphate. Okay, and that gives me just here's now the amino acid glutamine. Okay, and so you will have in your tissues ammonia that is made in your tissues will either be used by glutamate dehydrogenase to make glutamate or picked up by glutamine synthase, and one of the most abundant amino acids in your blood is glutamine because it's a major nitrogen carrier from breakdown amino acid. You can also recover that. The enzyme is glutaminase. Okay, so glutaminase basically, I need to redraw all this, but here, this would be this side chain here on the glutamine, and effectively just water come along, leave off the ammonia, right? And that'll give you, I'm not going to draw the structure, but that'll give you glutamate. So you can see here, making glutamine, one pathway, one enzyme requires energy input. Breaking down glutamine, other direction doesn't require energy input. Fits with all the stuff we talked about, right? Pathway in one direction can't be the same as a pathway in the other direction. One direction will release energy, one direction doesn't, so this all fits all the thermodynamic stuff I showed you before, but also shows how you can use basically glutamate, as an additional way to carry nitrogen around. And it turns out, you'll see when we talk about nucleic acid metabolism, you sometimes also use glutamine as a nitrogen donor for nucleic acid synthesis. All right, so now you as an animal, you eat food, you break down the protein from the food you eat. That protein, you'll oxidize the carbon, you end up generating some ammonia, and that ammonia is picked up in your tissues by either glutamate dehydrogenase or glutamine synthase, and then you send off that glutamate and glutamine in your blood that ends up in the liver or kidneys, and then the liver or kidneys' job is then take that, recapture that ammonia, turn it into urea, and put it out in the urine. Okay? So glutamine and glutamate end up being really important amino acids, particularly glutamate, in terms of how you're taking nitrogen into the system and out of the system. And at least for humans, glutamine is a way to carry nitrogen to get it to the tissues that make urea so that you can generate urine and get rid of nitrogen in your body. Okay? All right. So this puts it into the system, but you have all these other amino acids that are out there. And so it turns out the way that once you get the nitrogen into glutamate, you can now use glutamate and alpha-ketoglutarate to effectively exchange amino acids with alpha-keto acids. Okay, so here's this is oxaloacetate. Okay. Oxaloacetate is an alpha keto acid. Okay. Alpha to the carbon-ketone group, alpha to the carboxylic acid. Here is the amino acid aspartate. Okay. So amino acid is the amino acid right? So if I do just like you saw in the malate aspartate shuttle, start here with glutamate and make an alpha ketoglutarate, okay? This way, the glutamate over here, amino acid, alpha ketoglutarate is the alpha keto acid. All right, this is the reaction you saw in the malate aspartate shuttle. All right, this here is completely balanced. Okay, the amino acid is more reduced than the alpha keto acid, but my electrons are conserved because as I'm doing the reduction here, I'm doing the oxidation of glutamate to alpha ketoglutarate. Nitrogen is conserved. There's a nitrogen here, there's a nitrogen there. So if I draw this whole thing out, all electrons, all nitrogens, all carbons, all oxygens, everything here is conserved. I'm basically swapping the alpha keto group for the amino group. And it turns out this is the route cells use to transfer nitrogens around to make all kinds of amino acids and interconvert them with their alpha keto acids. Okay? So if I have alanine, okay, the amino acid, and turn it into pyruvate, the alpha keto acid. But to do that, I give the nitrogen to alpha ketoglutarate and make glutamate. Now I have pyruvate. That glutamate I can use and go to alpha ketoglutarate by giving nitrogen to another alpha keto acid like oxaloacetate and make aspartate. All right? Do this over and over again with any amino acid and any alpha keto acid. And effectively, once I put the ammonia into the system with glutamate dehydrogenase, now it's on glutamate, I can now do these exchanges between other amino acids and other alpha keto acids. So if I'm doing catabolism, right, I want to catabolize my aspartate, well, I basically give the nitrogen to alpha ketoglutarate and I make glutamate, and then glutamate can participate in that reversible glutamate dehydrogenase reaction, and that's how I can get ammonia. If I want to make aspartate, well, I put ammonia into glutamate by the glutamate dehydrogenase reaction, and now I can make aspartate. So catabolism, sending all my nitrogen to glutamate, where I can use glutamate dehydrogenase to make ammonia. If I want to do anabolism, take all of my nitrogen in through glutamate dehydrogenase and swap it around with all the other alpha keto acids to make amino acids. And this is effectively, in a nutshell, how you do amino acid synthesis and breakdown. Now there's a couple very notable exceptions to this that I want to just point out. The first one is glutamine. I drew right there in the middle board. That's how you deal with glutamine. Okay, so it's basically glutamate plus minus ammonia. All right. Same thing with asparagine. N is asparagine. All right. This works just like glutamine, except if you look at asparagine, asparagine is basically glutamine but one carbon shorter. So that's aspartate going to asparagine. So very similar thing. Why nature chose glutamine and glutamate to be the donor and not aspartate and asparagine, I have no idea, but that's what they are. Proline, I'll show you this next time. Proline is basically cyclized and reduced glutamate, right? So proline, I'll show that next class. We'll go through proline, that'll make more sense then. So just realize proline is different. Glycine, you make glycine from serine, okay? We're going to talk about serine metabolism a lot next time. So glycine and proline will come up next time. But the last one for today is, the other weird one is arginine. Personally, I always found arginine the hardest amino acid to remember and to draw. Right? It's this crazy amino acid with this crazy side chains with all these nitrogens in it, 3 nitrogens. Okay, it's this special amino acid with 3 nitrogens in the side chain, 4 nitrogens if you count the amino carbon. And it turns out arginine is urea metabolism, and so we'll talk about arginine a lot here end of today going into the next class. But before we get to that, I want to talk a bit about this reaction, this swaps right here. So it turns out this reaction here has a name. This is called transamination. And the enzymes that carry out this transamination, which basically, other than these exceptions, is how you synthesize and break down all the other amino acids, are called transaminases. All right? So effectively, a transaminase has selectivity for a specific amino acid or set of related amino acids in some cases, and all of them interconvert the alpha keto acid and the amino acid with glutamate and alpha ketoglutarate. This exact reaction right here. So there is a specific transaminase that carries out this reaction called AST, aspartate transaminase, that basically has specificity for aspartate and oxaloacetate and does the transamination reaction with glutamate and alpha ketoglutarate. All right, there's another one of these that's quite famous, ALT. That's alanine transaminase. That's basically this reaction right here, specificity for alanine and pyruvate, does the transamination with glutamate and alpha ketoglutarate. Why do I point out AST and ALT? Because those of you who are going to go to medical school, you will find if you do a blood test for liver, okay, your so-called liver function test, what do they measure? They measure these 2 enzymes that are very abundant in the liver, that are basically, if these leak into your blood, that's what happens. You damage your liver, liver leaks excess levels of these transaminases into your blood, that's a sign of liver damage. Now, all transaminases are completely active and reversible. All right, these are some of the most active enzymes you have in your body, and they're completely reversible. So you will basically drive these reactions in the direction that basically cellular demand wants. Right, delta G is very close. Delta G naught prime is very close to 0. So you basically have your alpha keto acid will interconvert your amino acid based on the transaminase, and ultimately go between glutamate and alpha ketoglutarate, and then that glutamate will go to alpha ketoglutarate here, which requires to effectively take ammonia in and out of the system. All right? So glutamate dehydrogenase and the transaminases will work together. So this direction, that's catabolism. All right? You want to get energy from your amino acid, you give the nitrogen to alpha ketoglutarate, now you get the alpha keto acid, you can burn that. This makes glutamate, the glutamate makes the alpha ketoglutarate back, which gives you ammonia, and now just keep running a cycle and dump nitrogen. If I want to do anabolism, okay, I can take nitrogen in from alpha ketoglutarate, make glutamate, transaminate that nitrogen onto an alpha keto acid, now I get an amino acid. So if you're building muscle, you're pulling amino acids out. Anabolism, if you're starving and breaking down muscle, you're pulling amino acids this way, making ammonia. Completely reversed. All right, let's take the break, and then after the break, we'll talk about in detail transamination reactions, and then hopefully get to nitrogen or arginine and urea. All right, 5 minutes or 4 to 5 minutes. About transamination reactions, and transamination reactions is a class of reactions that requires a cofactor, like all these fancy things, doing this swaps between alpha-keto groups and amino groups. Glutamate and alpha-ketoglutarate requires a cofactor, and the cofactor comes from a vitamin. The vitamin in this case here is vitamin B6 that was put in your cereal, okay? And then your cells take the vitamin B6 and basically add a phosphate here, oxidize this alcohol to the aldehyde, and protonate the nitrogen, and that gives you this molecule here, pyridoxal phosphate, which is always abbreviated ELP. And basically, if you see pyridoxal phosphate written somewhere, pyridoxal phosphate is about amino acid metabolism, okay? Pyridoxal phosphate is required for transamination, so I'll take you through how transamination works. So here's just a generic amino acid that I drew here in purple. All right, here's our pyridoxal phosphate. Okay, I'm just gonna draw, I'm not gonna draw all the side chains and stuff like that, but effectively, this will participate in the following chemistry. Let me draw all every single piece here. So this is just that middle part there of the molecule. Here I use water. created the alpha keto acid. Because if you follow this through, it started with the amino acid, pyridoxal phosphate, and now I've gone to the alpha keto acid with the former nitrogen group here now on this cofactor intermediate. So we'll draw out 480. Okay, and this here is called PMP, or pyridoxamine phosphate. All right, and so effectively, what I've done is I've gone from the amino acid to the alpha keto acid, which is oxidation, but I oxidize something because alpha keto acid and amino acid, different redox states. Here the cofactor PMP is now in a different redox state. So PLP is more oxidized, PMP is more reduced, amino acid is more reduced, alpha keto acid is more oxidized. And so I've basically generated the alpha keto acid in a different version of the cofactor PMP. Now I can just reverse this whole thing, in this case now using a different amino acid, okay, or sorry, in a different alpha keto acid. Okay, so here's my alpha keto acid. I just reverse this whole thing. draw it out, but then you get PLP again, and what we've done is we've generated a new amino acid. Okay, so effectively, transamination, you're basically moving the nitrogen as well as 2 electrons onto PLP to make PMP, and then you're just taking the PMP, taking that nitrogen and 2 electrons, putting it onto the next alpha keto acid, regenerating PLP and a new amino acid. Very, very reversible reaction that effectively allows you to use this cofactor, PLP and PMP, to end up moving nitrogen between alpha keto acids and amino acids in a way that's conserved, right? You get an amino acid and an alpha keto acid as both substrates and products for the reaction. Now you'll see PLP is important for other reactions. I'll show you another one PLP is used for next time, involved in serine and glycine metabolism. PLP is also required. I think I promised last time, I'm not going to go through the mechanism, we don't have time, but here's the amino acid serine. Okay, we're going to talk about serine a lot. Okay, you can also use PLP to effectively decarboxylate serine, so take that CO2 off serine, and then this gives you this molecule, which is ethanolamine, which of course is required for phosphatidylethanolamin, for alcohol here that you can now esterify a fat to, and that's or a phosphatidic acid to, and give you phosphatidylethanolamin. Okay, so PLP ends up being this really useful cofactor for this. All right, so if you're a fish and you live in the ocean, all you would do to catabolize an amino acid is you basically do this transamination, ultimately to get the nitrogen onto glutamate, and then once it's on glutamate, you can then run glutamate dehydrogenase to make ammonia, which just floats off into the ocean. If you're us, if you're a mammal or a bird, you do the same thing. You transaminate the glutamate, or you put it onto glutamine, and then ultimately you have to carry that to our livers and our kidneys, which end up making urea, or if you're a bird or a reptile, uric acid, which we'll cover on Monday. But how you make urea is really effectively arginine metabolism. Okay? So all organisms make arginine, and so I'm going to show you how you make arginine. But if you make arginine and you break down arginine, you're going to produce urea. And so this is really where urea comes from and why you do urea the way you do it. And so just to be explicit about this, the source of urea is arginine. So let me remind you what arginine looks like. So this here is arginine. All right? And if I come in with water, and carry out that reaction, what I get is urea. So making urea is effectively taking that part of the side chain off of nitrogen, and then what I'm left with is this other amino acid, which is not found in protein. Okay, but this is also, if you look at it, it's an amino acid, it just has a side chain not found in protein. This is the amino acid ornithine. And so effectively, if I can make arginine, I can take the arginine, release urea from the arginine by the reaction I just saw you. I get ornithine, and then if I can take my ornithine and regenerate an arginine, which is basically adding 2 amino groups and a CO2, that is a cycle I can run that now allows me to excrete urea. And so, indeed, the way that we make urea is a cycle called the urea cycle, which effectively has to do with arginine metabolism, where effectively we just cycle between making ornithine, making arginine, releasing the urea, get an ornithine again, go back. Cycle, just like the CCA cycle. In fact, this was discovered by our old friend Hans Krebs here from the Krebs cycle, except Krebs discovered the urea cycle first. He discovered it in 1932. So he had cycles on the mind, which kind of helped him discover by the Krebs cycle later, as he first described the urea cycle. It was the first metabolic cycle described. By the way, I took this specific picture. This is hanging outside the men's room at the Royal Society in London, which I found to be pretty funny. It's kind of British humor, right? You discover the urea cycle figure and hanging outside the men's room. Anyway, it's kind of funny. So our British colleagues at the Royal Society have a sense of humor. All right, the urea cycle involves compartmentalized metabolism. Okay, and so what does that mean? That means that it's actually occurring in different locations here in the cell. So first here I'll just start by giving you an overview of the urea cycle. So here we have arginine. All right, as I just showed you, if I have arginine, I can actually take water and take that side chain, the reaction shown over there, and generate a molecule of urea, all right? And what I'm left with is ornithine, okay? So it turns out ornithine, this happens in the cytosol, but ornithine in a reaction that occurs here in the mitochondria will generate another amino acid not found in proteins called citrulline. All right, to do this, it's basically going to pick up CO2 and an ammonia in the mitochondria, make citrulline, all right? Citrulline, it was originally isolated from watermelons, all right? So, because it was originally isolated from watermelons, it was called citrulline. And then citrulline will be used to generate a molecule called arginosuccinate, which will then generate arginine, and via a complex set of reactions that I may not get to today, this is going to incorporate the other nitrogen as ammonia, although it's not direct like it is there. Okay, so let's at least start going through how this works. And since I only have 6 minutes left, we may have to finish it in the next time, but we at least have time to discuss the first of these, the mitochondrial reactions. All right, so we're going to add CO2 and ammonia to ornithine. And so the way this happens is kind of sort of chemistry that you've seen before. It's a little different. Remember, CO2 is in equilibrium with bicarbonate, okay? And So I can come along and I can phosphorylate bicarbonate just like when we've done other carboxylation reactions. So that gives us this phosphorylated bicarbonate. And then when we did carboxylation reactions, normally this would interact with biotin, except in this case, rather than reacting with biotin, there's an enzyme that will have it react with ammonia. Lose the phosphate. And that gives us this molecule, which is called carbamic acid. And then carbamic acid can come along and be phosphorylated, plus another ATP, and that gives us this molecule, which is called carbamoyl phosphate. All right, so carbamoyl phosphate, this entire set of reactions, basically CO2 and ammonia, basically 2 ATP, everything that I drew here is catalyzed by an enzyme called CPS, or carbamoyl phosphate synthase. Names of the enzymes are not that important, but carbamoyl phosphate is what's generated in the mitochondria. So conditions in the mitochondria help you generate carbamoyl phosphate. And then that carbamoyl phosphate will react with ornithine. Okay, so there's this here is ornithine. Lose the phosphate. And this here, so carbamoyl phosphate plus ornithine gives me this. This is citrulline. This reaction occurs in the mitochondria. Basically, make carbamoyl phosphate with CPS and then react that to make citrulline in the mitochondria. And then that citrulline will go out to the cytosol, and a series of reactions that I don't have time to tell you about in 2 minutes, I'm not even going to try. You'd end up generating arginosuccinate. Ultimately, that arginosuccinate can lead to add another nitrogen. You get arginine, and then arginine can release urea, regenerating ornithine. Once I make the ornithine again, the ornithine can come back over here, pick up another carbamoyl phosphate in the mitochondria, make a citrulline, pick up another nitrogen to make arginosuccinate, and then arginine, etc. Cycle that ultimately allows you to pick up ammonias and excrete that nitrogen. Okay, next time we'll pick up talking about the rest of it and show you how it